Hello, students. In this screencast, we are going to be doing a little bit of drawing, believe it or not. I'll kind of cut away for a moment to draw on a diagram. And to access that diagram, you're going to want to go to Schoology. And in the nervous system folder, you'll see notes and study guide. And here we have the study guide for the nervous system unit 2. So if you click on that, it brings you to this document. And if you have printing capabilities, this would be a really useful diagram to have. I'm going to kind of draw on this. If you don't have printing capabilities, that's okay. You could just kind of rough sketch this, just kind of draw a circle and some lines, and that will work just as well. For our next screencast, I'll just kind of point out this diagram will also be useful as well. So if you're at the printer, might as well click both of those and this one as well. So we're going to do some drawing. So let's get to it. Last time we were focusing on the different parts of the brain and we looked at their structures and what those structures all did. This time we're going to get into the much more interesting, in my opinion, question of, well, how is it that the nervous system actually sends information around the body? So if my brain is sending a signal to my arm, how is it doing that? What's actually happening at the cellular and molecular level to get at that, to let that happen? So that's what we're going to do. So to understand that, we need to understand the cells of the nervous system. And the cells of the nervous system are called neurons. And here's a lovely diagram of the structure of a neuron. And I think it's worth taking the time to just draw that out. If you're a terrible drawer like myself, you can go ahead and just kind of do this sort of weird thing that I kind of created, and you can kind of see a parallel between these two, I think. But we're going to kind of label those parts as we go. So here are the parts, and I'd pause, I would draw, and then I'll kind of point out what they are. So the first structure is the cell body. That's this kind of circular area that contains the nucleus. Last semester or last year in biology, you learned all about cells and all the organelles and things like that. They're largely in this cell body. This structure isn't particularly important for the processes we're about to discuss, but it's there. The dendrite are all these branches coming off of the cell body, these many small ones. Their job is to receive signals from neighboring cells. So again, if we're sending messages through our body, like in a chain, these are receiving the signal from a neighboring cell. The axon then is this long extension right here. So it'd be this one right here, axon. And the axon's job is to relay the signal to the end of the cell. At the end of the cell, we get to what is called the synaptic terminal. And the synaptic terminal here is what relays the signal to the next cell. So you can kind of see there's this directionality to the neuron. The dendrites receive the signal, the axon relays it to the end of the cell, and then the synaptic terminal is going to relay it to the next cell. In this case, these kind of weird brown things are muscle cells. So this neuron is contacting a muscle. Couple more things. Wrapped around the axon are these little sausage-length things. This is the myelin sheath, a fatty layer that insulates the neuron and basically increases the speed of transmission. We talked about that very briefly when we looked at the brain structures. The myelin is what gives white matter its white color, and not all neurons have it. And then there's these little gaps in the myelin sheath. Those gaps are important. Those gaps are called the nodes of Homvier. Or you can pronounce it, Reinvier, whatever you like. But these are these little gaps in the myelin sheath, and it turns out those gaps are really important for the myelin sheath to do what it needs to do. So these are the parts of the neuron. So there they are with the labels. Everything's good. All right. I also want to point out not all neurons look the same. So you can see a variety of neurons. They generally have the same structures. This one, for example, does not have the myelin sheath. So this would be a gray matter neuron. It has a lot more branches, so there's variety. But in general, those structures I pointed out are common to all. So what we're going to do now is we're going to look at nerve signals. Again, how do we kind of send messages throughout the nervous system? And this is a complicated process, so you're going to have to stay focused, and there's a lot of pieces that I kind of put in place, and then those pieces come together. So just kind of bear with me for a little while, and if it seems like I'm just throwing random terms at you, it will come together, I promise. But you're going to have to stay focused on this one. So to understand how a cell sends a message, we have to understand something called a resting potential. Cells in the nervous system, neurons, have what is called a resting potential, which means there's a difference in charge across the membrane. Think potential energy. If you lift a ball in the air, there's a potential energy there. So a resting potential is similar to that, only instead of moving a ball away from the ground, in this case, you've got a difference in charge. There might be a slight positive area inside the cell and a slight negative area outside, and that difference in charge is that potential energy. So to understand what contributes and what causes this resting potential, we need to look at these charged particles called ions. And you may remember that term from chemistry. Sodium and potassium are two ions that are really important in contributing to the resting potential, this difference in charge across the membrane. So take a look. Sodium ions are purple, potassium ions are green. And just look at this diagram. Here's the membrane that separates the outside of the cell from the inside of the cell. Maybe you remember this structure from biology last year, those phospholipid bilayer. Remember that? I don't know. These are little proteins in there. So is there a greater concentration of sodium inside the cell or outside the cell? What about potassium? For sure, hit pause, reflect, look at the picture, figure it out, and then continue the video. Make sure you're understanding and following along. If you look at these pictures, you should see that there is actually more sodium outside the cell than inside. So there's a greater concentration outside the cell than inside of sodium. For potassium, it's the opposite. There's a greater concentration of potassium inside the cell than outside. And so this contributes to that difference in charge across the membrane. So what I want to do is I want to just start a sketch. So I'm going to switch to this diagram, and I'm going to draw on that, and I'll be back in a moment.
All right, so you should have this printed off, or you could just do a rough sketch of this neuron. It doesn't have to be perfect. Now what I'm going to do is I'm going to zoom in on a little area of this axon. So I'm just going to kind of draw a box around this little area right here. I'm going to blow it up. So I'm going to kind of blow it up like that. And then you see these two lines inside. I'm just going to redraw those two lines larger so we can kind of see what's going on in there. So, you know, this is inside the cell, and then this is outside the cell. So these two lines, we get it? Okay, good. Now, we're gonna look at the concentration of those different ions that we talked about before. So I'm gonna have sodium ions. I'm gonna use a red marker to represent those, a red pencil here. And I'm just gonna represent those as little circles. And then I'm gonna use a blue pencil to represent potassium ions, and those are going to be little triangles. So from that previous slide, we said that there's a greater concentration of sodium ions outside the cell. So I'm just gonna make a bunch of red circles, kind of a lot of them, outside the cell. And there's a handful of them inside, but there's a greater concentration outside the cell. Now, hopefully from biology last year, you remember that when there's a difference in concentration, you have more of something on one side than the other. There's this tendency to equalize. So these sort of, quote, want to go inside the cell to balance out the number, but they can't. These charged particles aren't able to cross the membrane. So they're kind of stuck for now. Potassium ions then, the concentration was greater inside the cell. So I'm going to make a bunch of blue triangles inside the cell. And then there's a handful outside. But again, greater concentration. So the sodium ions tend to want to go into the cell. The potassium ions will tend to want to go outside the cell. But no one is able to move right now because the membrane doesn't let them cross. Now there's one final component I want to mention. I'm just going to draw kind of one black box here. That black box represent proteins. So those are proteins, and it turns out these have kind of a negative charge to them. And so those aren't terribly important for the overall process, but I just wanted to kind of mention that that also contributes to the charge. So that's kind of our scenario.
So to summarize, at rest, there's a greater concentration of sodium outside the cell, there's a greater concentration of potassium inside the cell, and there are also negatively charged proteins inside the cell as well that aren't really shown on this diagram, but they are there. So that is what's contributing to that resting potential. Now, we could actually draw a graph of this process. So let's take a moment and do that on our same diagram.
So we're going to draw a graph just down here in the corner here, and I'm going to make the y-axis and the x-axis kind of like that. And this graph is going to be of membrane potential. And the units are in millivolts, MV, versus time. And so basically what membrane potential is it's looking at what is the difference in charge across the membrane. So if we were able to say, put a little needle in here, and actually this is what scientists do. They take giant neurons and they use tiny needles. And if you put one little needle in here and one little needle in there, and then you measure, like, is the charge equal across them or is it different? It turns out that in neurons, it's different, and we call that difference a resting potential. So if you add up all the positive and negative charges inside the cell and all the positive and negative charges outside the cell, and you compare them to each other, the inside is more negative than the outside. And we can quantify that with this value, membrane potential. And it turns out that at rest, the cell has a resting potential of minus 70 millivolts. So when a neuron is just chillaxing, sitting around doing absolutely nothing, because there's different concentrations of these charged particles, that means that the inside is minus 70 millivolts with respect to the outside, and it kind of is this, it just kind of sits at minus 70 over time. That is what we call resting potential.
Okay, so if you want a prettier diagram, here it is. So at rest, because there's a greater concentration of negatively charged proteins inside the cell, the resting potential is minus 70. So basically, there's more negative charge inside the cell than outside. That difference in charge creates kind of a potential energy. It's a bit like, think of the ground and a ball. There's this potential energy there, and the ball will tend to drop. Only in this case, it's a positive versus negative charge. There's a negative charge inside the cell, there's a positive charge outside the cell, and so there's that potential energy there. So if we measure the value of that difference, it's minus 70 millivolts, and a cell at rest will just be like, boop, at minus 70, just chillaxing. So if your neurons are just chillaxing, they're at minus 70, and that is the resting potential. What I want to do in our next part is look at, well, how do we take advantage of this resting potential to make something interesting happen? So I'm going to pause and we'll continue this video with part 2